where myosin will need to bind when they interact. So to keep you from moving, troponin and tropomyosin block the myosin binding sites on actin. And when you want to move, those two proteins move away from the myosin binding sites and allow them to interact. So we'll talk about how that's going to occur again when we get to the physiology. Right now, we're just letting you know that these things exist. And like usual, these are not the only proteins. There are lots of other proteins, especially structural proteins, helping to hold all these things together. You do not need to know these structural proteins in the box. You do not need to know titin, nebulin, alpha-actin, myomycin, or dystrophin. You do not need to know those structural proteins. But that's pretty much it on the microscopic anatomy of important structures in the muscle cell. Remember, like usual, trust the exam review. And so the rest of this chapter is switching in finally into the physiology. We're going to talk about how do you move. And like usual, there's going to be lots of steps in this, just like how there were lots of steps in the generation of an action potential. There are going to be lots of steps involved with how do you generate a muscle contraction? How do you move? And I want you to know the steps in order for the generation of a muscle contraction. How do you move? Excuse me. And we're going to go through them. And just to help you to save some time, a lot of these steps are highlighted already in the slide on your PowerPoint. But I'm just going to go in order of the slides that are here. And unfortunately, in this order, which is the order it's given in your textbook, they actually don't start in the beginning. They actually start towards the end. And they start off basically when you're in the process of moving. Remember, when you're moving, you're really just shortening the muscle. And you're shortening the muscle by shortening the cells. And you remember the cells are shortening because the myofibril is shortening. And you remember the myofibril is shortening really because sarcomeres are shortening. And you know now sarcomeres are shortening because actin and myosin are interacting. So we're starting here. We're starting where actin and myosin interact. And we help to explain how actin and myosin will interact in what we call the cross-bridge cycle, or the sliding filament model or mechanism. It's called the sliding filament model because they're basically looking like they're sliding across each other, kind of like how your fingers look when you interlock them. If you were to slide them to interlock your fingers, that's a lot like how the sliding filament model of how actin and myosin acts will represent. How so? Well, it's really because myosin is pulling on actin, like a game of tug of war. Myosin is going to actually pull actin towards the M line, towards the middle. And as it's pulling on actin, actin will pull whatever it's attached to. And remember, actin's pulled or attached to the Z disc. So when actin's being pulled by myosin, one thing we're going to see when we look at it is that the Z discs will actually look like they're being pulled towards each other on either side of the sarcomere. It's really because they're being dragged because they're attached to the actin, which is being dragged towards the middle of the cell. But again, keep it simple. We're going to go through the steps. It's really four major steps that help to explain the sliding filament model. And again, not only is actin and myosin going to move, but what's attached. And I told you, we're going to see this in the sarcomere. And you can see that on this picture here. This picture is just showing us all the changes we're going to see within a sarcomere as the muscle, the sarcomere, the myofibril contracts. And again, just think about what everything's attached to. I told you myosin's going to pull on actin, and actin's attached to the Z disc. So one thing that happens in a sarcomere as you contract is that the Z discs will move towards each other. They will move closer together. That's one thing that happens in your sarcomere during a contraction. Z discs move closer together. What else? Well, as myosin is pulling on actin, they're going to begin to overlap. Remember, they're sliding across each other. Why is that important? Well, you remember there are areas in your sarcomere where there is no overlap, where there's only thin filaments or only thick filaments. Well, we're overlapping now, so you're going to begin to lose some of those areas. Remember, one area was called the I band. Remember, the I band is where you only have thin filaments. On this cartoon picture, it's represented by the light blue box. That light blue box is representing the area we call the I band. And as you contract, well, myosin and actin are going to slide and overlap, so you're going to lose where you only have thin filaments. So during your contractions in a sarcomere, we say the I band will narrow and possibly disappear. Why? Because myosin and actin are overlapping. Remember, there was also an area where we only had the thick filament, myosin. Remember, that was called the H zone. Well, again, as you're contracting, myosin and actin are overlapping. They're sliding against each other. So you're going to also lose that area of where you only have thick filaments. So just like the I band, the H zone, when you contract, will narrow and also possibly disappear. Those are the three major things I want you to know that happen in a sarcomere during a contraction. One more time. Z discs move closer together. I bands will narrow and possibly disappear, and H zones will narrow and possibly disappear. That's what happens. What happens to the A band? Remember, the A band runs the length of myosin. And did I mention anything about myosin getting shorter? No. Well, the A band is going to stay the same. That's why I didn't mention the A band. Okay. So that's what happens in your sarcomere during a contraction. And how? How do actin and myosin interact? What's the cross-bridge cycle? What's the sliding filament model? Well, it's really just 4 steps. 4 simple steps. And these 4 steps are basically the last 4 steps in the generation of a muscle contraction. It's how you shorten. And you remember, first thing that actually has to occur is that you need to remove those regulatory proteins. So let's assume that myosin and troponin have already been moved. And how does that happen? It's super simple. It's calcium. To get troponin and tropomyosin to move, all you need is calcium to bind to troponin. And when calcium binds to troponin, troponin and tropomyosin will move away from the myosin binding sites on actin. Kind of think of troponin as the boss. So you only have to talk to troponin and it will tell tropomyosin what to do. So calcium will bind to troponin, causing troponin and tropomyosin to move away from myosin binding sites on actin. So we're going to assume those steps have already occurred. So these 4 steps I'm about to mention are this cycle, this cross-bridge cycle, this sliding filament model. First step is something that myosin needs to do. If you notice from the picture, you see the yellow beads up top. Those are representing actin. And this long red rod-like structure with these bulbous heads coming off is myosin. And it looks like actin is kind of hovering over myosin. And those little bulbous structures, what we call myosin heads, they need to look up at actin. They need to cock their heads back. And in order to do that, they need to use ATP. When you use ATP, we say you hydrolyze ATP. So the first step is that myosin heads hydrolyze ATP to form the cocked position. AKA, myosin heads will use ATP to cock their heads back to look up at actin. Then once they do that, it's time to move on to step 2, which is for myosin to bind to actin. They need to touch. Remember, they're going to pull on actin in a game of tug of war. And in order for you to pull, well, you need to attach. So step 2 is that the myosin heads will bind to actin. And when the myosin heads bind to actin, they form a bond, which we call a cross bridge. So that's step 2. Myosin heads bind to actin, forming a cross bridge. And then once they bind, now they could pull. And when myosin heads pull on actin, we call that the power stroke. And you remember, they're going to pull them towards the center of the sarcomere. They're going to pull them towards the M line. That is step 3. In step 3, myosin heads pull actin towards the M line during the power stroke. But again, this is like a game of tug of war. You can't pull the rope once and win the game. You got to pull, then let go of the rope with one hand, then reattach to it, then pull again, then let go with one hand, then reattach and pull again. Your myosin heads need to do the same thing. Once they pull once on actin, they need to let go so they could grab and pull again. So the last step, the fourth step, is how the myosin heads are going to let go of actin. And it's super simple. To release actin, myosin heads simply bind ATP. That's it. They don't even use it. They just bind to it. And when myosin heads bind to ATP, that will cause them to release actin. And then this cycle will resume again. That ATP the myosin heads bound to release actin is actually the ATP they're going to use or hydrolyze to form the cocked position. And this cycle will occur multiple times until you've reached maximum contraction, until you've flexed or contracted the muscle, its maximum possibility. So those are the steps. They happen over and over until you hit maximum muscle contraction. One more time. Myosin heads hydrolyze ATP to form the cocked position. Then they bind actin, forming the cross bridge. Then they pull actin towards the M line in the power stroke. And then they bind ATP to release actin. Then repeat. Myosin heads hydrolyze that ATP to form the cocked position, bind actin to form the cross bridge, pull actin towards the M line in the power stroke, bind another ATP to release it. Then they will use the ATP that form the cocked position, bind to actin to form the cross bridge, pull actin towards the M line to form the power stroke, and then bind another ATP to release it, and repeat over and over again until you've hit maximum contraction. So this is basically how your cells are contracting, how the myofibrils in the cells are contracting, the cells themselves are contracting, the muscle is contracting, and that's really how you're moving. These are the last 4 basic steps to a contraction. But again, there's more to know. I told you those were the last 4 steps. So we got to go way back to the beginning. And it actually begins with nervous tissue. You know, if you want to walk to the door, it begins with you deciding to walk to the door. And once you decide, it's going to begin with your brain sending signals via neurons to the neurons in your spinal cord, which will send that information to neurons and nerves in your body, motor neurons, which will then talk to the muscle cells. And remember, one way your neurons are communicating is via electricity. So it's really some sparks of electricity that will lead to an eventual contraction. It's really these excitable cells. Remember, neurons are excitable cells. They can make an action potential. Turns out, once a muscle cell gets a signal from a neuron to contract, it will also make a spark of electricity. Remember, this is why we did nervous tissue first. Remember, as soon as your neuron releases its neurotransmitter, you could get either an excitatory postsynaptic potential in the recipient cell or an inhibitory postsynaptic potential in the recipient cell. Well, we're talking about muscles. We're talking about stimulating them for movement. So your muscle cell, once it gets a signal from a neuron, will generate an excitatory postsynaptic potential, which will cause it to depolarize. And if it gets a strong enough signal, it will hit threshold and make its own action potential. Pretty similar to how a neuron does. So not only are neurons excitable cells, so are muscle cells. And we're going to see as we go through these steps in the generation of a muscle contraction, your muscle cells at one point will make electricity. So we name this combination between your cells making electricity, which will eventually lead to your actin and myosin sliding across each other. We call this the Excitation-Contraction Coupling theory. It's how we're going to talk about electricity and relate that somehow to this contraction, or this coupling of a contraction in your muscle cells. And when we talk about muscle cells and this coupling, like usual, there's going to be an optimum for everything. As in biology, there's optimums for when and how you can contract. And it has to do with the sarcomere. We saw in the sarcomere, there were lots of these random areas. Things were spaced out in a sarcomere when you're relaxed. And as you're contracting, things get shorter and squeezed tightly. There's a reason for this separation initially. It's because there's a relationship between the length of your muscles and cells and how much force you could generate. Turns out this is what we call the length tension relationship.